Okay, yeah, basically blood samples are taken, and then you got measurements of proteins in everyone's blood plasma. What are some of the basic headline results here in terms of how this protein data set tracked with biological age?
The headlines are a few. The first is that, using blood proteins, we can make a proteomic clock just as accurately as a methylation clock.
Mhm.
But that too, so that's known, but proof of concept that we can use large-scale proteomics data to achieve a similar thing. And I will say we just tried a sort of first-generation clock here. So just take all the blood proteins, put them in a model to predict age. Turns out, if you start from 3,000, you only need 200 to actually estimate somebody's age. And you can estimate it just as well with 200 versus all
Yeah,
3,000.
yeah,
And you
that
can
makes
go all the
sense.
way
Like
down
some
to
proteins are
20.
going to, be very, very intimately involved in the aging process, basically.
Exactly. And what we even find is that you can go down to just 20 proteins, and that will still get you 95% of the accuracy of the
Oh
larger
wow.
model. So those 20 are very multidimensional and heterogeneous in what they are and what they do. But one of the headlines is we can capture aging from blood proteins, and you actually don't need thousands of proteins to do it.